Hello and welcome to Zoology 142 Online. This is the lecture on the immune system and immune response. So after you complete this lecture and do the assigned reading in your book, you should be able to recognize the difference between the nonspecific and specific or immune response. You should understand the two different types of immune response, that is one involving B cells, the humoral immunity, and one involving T cells or cellular immunity. You should also be able to identify and describe the cells responsible for immune response, that is our B and T cells, and also understand how immune cells distinguish between self cells and pathogens. And you should also remember how the process of immunocompetence is achieved, although we really tackled this in the previous lecture on the lymphatic system. And finally, you should be able to describe the structure and function of antibodies. In addition to these, you should also be able to tell me about some immune disorders and the difference between, let's say, the HIV infection and autoimmune diseases. They are very different, and we'll cover those towards the end of the lecture. Okay, just a recap of some immune system vocabulary that we tackled in the previous lecture. The first of these terms is the term antigen. An antigen is any substance that elicits an immune response. Oftentimes we're talking just about a few proteins on the surface of a bacterium or a virus, so it doesn't have to be a whole organism, although oftentimes you will hear us say antigen in terms of talking about a whole organism. And so some antigens belong to pathogens. Pathogens are in fact disease-producing microbes, such as viruses, bacteria, fungi, and protozoa. Now remember that all pathogens are usually going to be antigenic, but not all antigens are actually from pathogens. For example, we look at people that are allergic to pollen, they will have an immune reaction to that pollen, even though the pollen itself is not pathogenic. Okay, finally the third term here is something called resistance. Resistance is the body's ability to ward off disease through both internal and external defenses. So as I said before, there are two major types of resistance. The nonspecific resistance, which is good against a wide variety of pathogens, and the specific resistance or immunity, which targets certain types of pathogenic cells and organisms. So we're going to talk about each of these in some detail. So the nonspecific resistance is present at birth, and it provides immediate but general protection against a wide variety of pathogens. And so nonspecific resistance is in itself subdivided into two categories: the external defenses, which are on the outside of the body, and the internal defenses, which obviously are on the inside of the body. So first let's take a look at some of the external defenses. I want you to imagine that a bacteria is drifting through the air as you're sitting here at your computer screen. And it's drifting until it sits on your skin. And of course this bacteria wants to get into your bloodstream and cause all kinds of nasty infections. So think about the different types of structures it has to permeate in order to get to the blood or other tissues inside the body. So the first thing it has to get through is the skin. And the skin is made up of three different tissue layers, right? We have an epidermis, a dermis, and a hypodermis. And the epidermis is made up of keratinized epithelial cells. And remember, these epithelial cells are in fact dead, at least the top layer is. And the cells are shed at an amazing rate. And so even if a bacteria gets onto those epithelial cells, unless it's able to burrow down in there and get through there very quickly, chances are that the bacteria will be lost just through shedding of epithelial cells. The other thing that is protective about skin is that it has a fairly acidic pH of around 4 to 5, and this can inhibit some types of bacterial growth. In addition, sebum, which is that oily secretion stuff that comes out of your sebaceous glands, can at times inhibit bacterial growth. Now this is not always the case. You've probably heard about people who have gotten infections of hair follicles, folliculitis, or sebaceous glands. And in particular, we do see people with large infections of sebaceous glands when they have cystic acne. And so sebum is kind of an equivocal defense against bacteria. On the other hand, perspiration contains a lot of nasty chemicals that again will inhibit bacterial growth. And these are things such as lysozyme, which is a bactericidal enzyme. If you remember back to Health 125, the suffix "cide" means to kill. So a bactericidal enzyme is an enzyme that can kill bacteria. Now, a bacteria or virus may not have to go through your skin in order to get into your bloodstream and your tissues. It could be inhaled or ingested. In this case, it's going to come in contact with the mucous membranes. And remember, mucous membranes line the internal surfaces of the mouth, the pharynx, the esophagus, the trachea, and so on. And so these mucous membranes are lined with mucosal tissue, that is, tissue that secretes mucus, and this mucus is moved around by cilia. And the purpose of this mucus is to trap particulate matter, including bacteria and viruses, and those cilia will eventually force that mucus up to the pharynx, where you will either spit it out or you'll swallow it. And once you swallow it, it's destined for the digestive tract, where we have powerful proteolytic enzymes that can sometimes kill or inactivate bacteria. So as I was saying in the previous slide, the digestive system does secrete some pretty nasty enzymes and also acid, which can inhibit bacterial growth and even kill some types of bacteria. Remember, the stomach is very acidic, pH of around 3, sometimes less than that, and it also has enzymes in there that will cleave and break down different types of proteins in the bacterial membranes. In addition, if you do get an infection of the GI tract, one of the symptoms will often be massive amounts of diarrhea. And this diarrhea actually has a beneficial purpose in that it helps to flush out the bacteria out of your digestive tract very quickly. And in this case, this mass peristalsis is stimulated by the presence of certain bacterial toxins or proteins within the GI tract. And occasionally diarrhea can become life-threatening. If you think about people with certain types of dysentery or cholera, the body's defense to get rid of this bacteria is to flush it all out. But while you're flushing it out, you're losing water, you're losing electrolytes, and so if we don't have adequate nutrition, that person can be very quickly become dehydrated, and this can be a life-threatening condition. Now, within the body's tissues and fluids, we also have a suite of antimicrobial proteins that are generalist in nature. And these include things such as interferons. Interferons interfere with viral replication, and they are produced by cells that have already become infected with a virus, and basically travel to adjacent cells that aren't infected and prevent those cells from becoming infected by messing around with or inhibiting viral replication. Another antimicrobial protein is something called the complement system, and I want you to read about this in your book. The complement system can actually help to eliminate bacteria and viruses from the body, and it also enhances the immune response of other portions of the immune system. So it will ramp up the B cells and T cells and also energize the phagocytes. And finally, there's a third type of protein called transferrins. Transferrins were transport proteins that help to transport raw materials such as iron. And iron and other raw materials are needed in order for bacteria to replicate. And so during times of infection, the transferrins will bind up all this raw materials and prevent its use for bacterial or viral replication. And of course, many of the white blood cells themselves are very generalist defenses. We're going to talk about our phagocytes, that is going to be our macrophages and also our neutrophils, but we also have something called the natural killer cells. And these are a certain type of lymphocyte that aren't as specific as the B cells or the T cells. And they tend to attack cells that display abnormal plasma proteins. So these could be invading pathogens, or sometimes it could even be tumor or cancer cells. And they kill these cells by releasing perforins or inducing apoptosis. Perforins are just compounds that literally perforate the cell membrane of the target cell and cause it to lyse or explode. And remember, apoptosis is programmed cell death. So natural killer cells can eliminate pathogens through the body by punching holes in their membrane or by causing them to undergo this programmed cell death known as apoptosis. Another nonspecific internal defense are our phagocytes. These are our neutrophils and also our macrophages. Remember, phago means to eat, and so these are large eating cells, and they ingest microbes and destroy them through the process of phagocytosis. So the neutrophils are a granular leukocyte, and remember, the neutrophils were the first responders to infections. So they're the ones that get there first and start phagocytizing bacteria, dead cells, cellular debris in the area, and after a while our macrophages will show up. Macrophages are derived from monocytes, and we have both wandering and fixed macrophages. Wandering macrophages travel throughout the body. They can travel through the bloodstream, undergo diapedesis, and get to the site of an infection. Whereas fixed macrophages, as the name implies, tend to be fixed in place. So we find these in areas which are extremely likely to harbor infection. For example, we'll find them in the subcutaneous tissues of the skin, and we'll also find them within the lungs, where we're breathing in lots of air that has particulate material in there and possibly lots of bacteria. So we tend to find fixed macrophages in areas where infection is likely. So now let's take a closer look at the process of phagocytosis. Phagocytosis begins when a phagocyte adheres to a pathogen or debris. The phagocyte will then form something called pseudopods, or fake feet. They grab hold of the pathogen and simultaneously engulf it in a vacuole, which we're going to call a phagosome. That phagosome will move internally into the inside of the cell, and it will merge with something called a lysosome to form a phagolysosome. Now, if you remember back from 141, we said that a lysosome was basically an intracellular stomach. That is, it's a small organelle that contains very powerful digestive enzymes, and it's used to digest foodstuffs within the cell, but it can also be used to digest and destroy bacteria, as it's doing here. So once those bacteria are digested by the lysosomal enzymes, we're going to have something called a residual body, which is just basically a lysosome that's done its work in digesting those bacteria. Finally, the phagocyte will use the process of exocytosis to get rid of any indigestible debris that results from that phagolysosome. Now, even though our neutrophils and our macrophages are very potent phagocytes, they can't actually destroy all types of bacteria. There are some types of bacteria that have proteins on the surface that make them impervious to digestion. And in fact, these bacterial cells can actually hide out and sometimes even divide within certain types of macrophages and also our neutrophils. And so our phagocytes aren't 100% effective against all types of bacteria. Another type of nonspecific internal defense is the process of inflammation, and you're probably very familiar with the process of inflammation. If you've ever poked yourself or gotten a mosquito bite or anything like that, you're going to notice one of the first things that happens is we're going to have hyperemia, that is a redness and swelling of the area caused by localized vasodilation. Remember, vasodilation means that our capillaries are basically dilating and becoming more permeable, and this permeability allows more of our phagocytes to move out of the veins, out of the capillaries, and into the surrounding tissues. So even within an hour of, let's say, an insect bite or an infection, we're going to start to see a massive increase in the numbers of neutrophils arriving at the area of injury. So let's take a look at this schematic that shows how inflammation and diapedesis helps to bring white blood cells to the area of injury and infection. So we start off with somebody who's accidentally stepped on a nail, and that nail probably has lots of bacteria on it, maybe even some virus particles on there, and other types of debris that we just don't want in the body. Now, the body's first response to this assault will be something called leukocytosis. That is an increase in the numbers of white blood cells. Specifically, we're going to be looking at an increase in the numbers of neutrophils. This is called leukocytosis. Once the neutrophils get close to the site of the injury or infection, they will begin to marginate. Margination is a process where neutrophils literally begin to slide along the walls and stick to the walls of the blood vessels. And basically it slows them down so they can undergo the process of diapedesis. So diapedesis is the process by which neutrophils are able to squeeze between the endothelial cells that make up the blood vessels, such as capillaries, and they move out in between these cells and enter the surrounding tissues. Now, they know where to go because of the process of chemotaxis. Remember that both damaged tissue and bacteria secrete certain types of proteins which these white blood cells can seek out. And so neutrophils in particular are kind of like bloodhounds in that they can sniff out the site of infection and travel to that area through the process of chemotaxis. And here we are at chemotaxis. Our neutrophils have migrated to the site of the infection by tracking the different amounts of bacterial proteins and also substances secreted by dead or dying cells. And once they get to the area of the infection, they're going to very quickly phagocytize any bacteria that are in the area and prevent that infection from spreading to different areas of the body. Now, if you want to see a rather corny video showing the process of diapedesis, click on the link below. Basically, it is a video made by a former medical student, and he sings along to the demonstration using the words of Garth Brooks and the Thunder Rolls. And it's a pretty corny presentation, but it's also pretty interesting, and it shows some great animation of the process of diapedesis. Okay, think back a couple of slides when we were looking at that puncture wound that got infected with bacteria. In most cases, our white blood cells are going to be very successful at eliminating all the bacteria in that area very, very quickly. But in some cases, we can get an infection that lasts for days and days. If you think about a staph infection that might cause boils on the skin, this infection is resistant to breakdown through our phagocytes, and so we get more and more white blood cells showing up in the area, and we eventually form an abscess. An abscess is basically a localized area of white blood cells that have been surrounded by connective tissue, because one of the body's last-ditch defenses to prevent spread of this infection is basically to wall it off. We have fibroblasts that will secrete a fibrous tissue capsule around the infected area so that the bacteria aren't able to migrate out of the area. And this is great because it does prevent the spread of the infection, but it also prevents the white blood cells from being reabsorbed by the body. And so you can get a very painful boil or abscess. And if you've ever had an abscess, you know that they are extremely painful, and in most situations the only way to deal with it is to go to the doctor's office and actually have them lance the abscess or boil. If it sounds disgusting, it really is. Here's a picture of somebody who's just had their boil lanced at the doctor's office. And sometimes you don't even need to have them lanced. Once they grow so large, they will rupture on their own. And they tend to be very, very painful up until the time that they rupture. Once they rupture, it's just complete relief. And I can say, as somebody who's had about 12 or 13 of these at one time, that they're very painful, and all you want to do is relieve the pain. So you go to the doctor's office, they'll probably just take a sharp scalpel and just make a punch into that abscess and drain it. Now, the pus coming out of here is not just liquid, it's actually very, very solid. And in some circumstances, they have to go in there and literally pull the stuff out with a Q-tip or with a fingertip. And so it's very painful, but you do feel a lot of relief once that abscess is drained. Okay, another general defense that the body uses to ward off infection is something called fever. And fever is an abnormally high body temperature. You know that the normal body temperature for humans is 98.6 or 37 degrees Celsius. And when we're infected with certain types of bacteria, our body will detect those bacterial toxins, which are called pyrogens, and it will elevate the body temperature. And remember, the hypothalamus is principally the organ of the brain responsible for adjusting our thermostat. And so these pyrogens act upon the thermostat and cause our body temperature to rise. And this is beneficial because it causes the liver and spleen to sequester iron and zinc, which are needed for microorganism replication, and it also helps to increase the metabolic rate, which can make immune response faster, and also it can make it faster for any repair of damaged tissues. Of course, fevers can get out of control, and in some situations the fever can be worse than the infection. So anytime you get a fever that is over, let's say, 104 degrees, you need to call the doctor's office or hospital right away, because that can be a life-threatening fever. Okay, so now that we've talked about all the different types of non-specific defenses, we're now going to focus on specific resistance, that is adaptive immunity. Adaptive immunity is the ability of the body to defend itself against specific pathogens. So in this case, we're not just defending ourselves against bacteria, but we're focusing on certain strains of bacteria. For example, a certain strain of the E. coli bacteria. And the adaptive defense differs from non-specific defense in three ways. First of all, specificity. It's specific against the pathogens that it detects and destroys. Second of all, it has memory. Once we encounter a certain type of pathogen, we will be better able to deal with that pathogen in the future. And that's something that is lacking in the non-specific defense. And the third difference is that the adaptive immunity is systemic. That is, even though we get a certain type of infection in the toe, if we get introduced to that pathogen again through another route in the body, we can respond to it more readily, even though we never had an infection in that area before. So we say that adaptive immunity is systemic or all over the body. So there are two different types of adaptive immunity. The first of these is called the cell-mediated immunity, which involves our T cells. And the second is called the humoral or antibody-mediated immunity, and this involves our B cells. As I was saying previously, cellular immunity involves our T lymphocytes, and these lymphocytes act directly against target cells by killing them. And usually we're talking about infected cells, because one of the things that T cells cannot do is detect bacteria or viruses that are floating free in body fluids. T lymphocytes can only detect endogenous antigens, that is, antigens that have been taken up by body cells either through infection or by antigen-presenting cells, which we'll talk about in a minute. So the big picture here is that the T lymphocytes are part of our cellular immunity, and they have cellular targets. And for the most part, these cellular targets are going to be infected body cells. On the other hand, our humoral immunity comes from our B lymphocytes. B lymphocytes don't deal directly with infected cells, but they do deal with antigens that are floating free throughout the body. And we oftentimes call these exogenous antigens because they're floating within the blood plasma or other fluids of the body. And the way that the B lymphocytes deal with these invading cells is they will secrete something called antibodies. And antibodies are special antimicrobial proteins which are produced by plasma cells, a certain type of B cell, and they bind temporarily to the target cell, whether it's a bacteria or a virus, and they will inactivate that cell and mark it for destruction by the nonspecific immune defense, such as our phagocytes. So the big picture here is that humoral immunity involves antibodies, and the targets are extracellular. That is, they are not our own body cells or infected cells. They are in fact free-floating bacteria and viruses. So just a reminder about who our lymphocytes are. Remember that T cells or T lymphocytes mature and undergo immunocompetence in the thymus, and they can only recognize endogenous antigens. That is, they can only recognize antigens which have infected a body cell or been bound to by something called an antigen-presenting cell. And so they participate in the cell-mediated immune response, and they eliminate pathogens once they infect the body cell. The B cells, on the other hand, go through immunocompetence in the red bone marrow, and they can recognize exogenous antigens floating free in the bloodstream and to some extent endogenous antigens. And they participate in the humoral immunity or antibody-mediated immune response. That is, they destroy pathogens remotely by secreting antibodies, and these antibodies help to inactivate and eliminate these pathogens from the body. Once the lymphocytes are created, they're then going to need to go through the process of immunocompetence. Immunocompetence, if you remember, is the process whereby these lymphocytes learn which targets to attack and which targets not to attack. And so immunocompetence involves learning that your own tissue should not be reacted to, but that foreign tissues should be reacted to. And so immunocompetence of the T cells happens in the thymus, whereas immunocompetence of the B cells happens in the red bone marrow. Now, once our B and T cells undergo immunocompetence, they are then going to move out of our primary lymphatic organs into the secondary lymphatic organs. And these include our lymph nodes, our spleen, our Peyer's patches, and also the MALT or mucosa-associated lymphatic tissue. And basically these areas are the sites where our lymphocytes are actually going to encounter antigens and destroy them. So these are our battleground tissues. So within these battleground tissues or secondary lymphatic organs, our lymphocytes will come in contact with the antigens. And once they come in contact with their specific antigen, they will become activated. So once those lymphocytes are activated by the respective antigens, they will undergo the process of proliferation and differentiation. That is, activated lymphocytes will divide many times to make clones of themselves. And these clones will then differentiate into one of 3 or 4 different types of cells. For example, both B and T cells have memory cells that can help us to remember an infection, and they have a long lifetime in the body. They will also differentiate into effector cells. The effector cells are things such as the cytotoxic T cell and the plasma cells. These are the cells that actually do the dirty work. For example, the plasma cells secrete antibodies, and the cytotoxic T cells are going to go around killing and eliminating infected body cells. Now, before we go on to talk about the way that B and T cells eliminate antigens from the body, we need to go back and review our definition of antigens. Remember, an antigen is any substance that elicits an immune response. That is, it ramps up our B cells and our T cells. Usually an antigen is actually just a portion of an organism, usually a glycoprotein. So these are large, complex molecules that are not normally found in the body. So they are not self-proteins, but these are foreign proteins. So we have both complete antigens and something called haptens. Complete antigens, as the name implies, have full immunogenicity and reactivity. By immunogenicity, we mean they are able to stimulate proliferation of specific lymphocytes, that is, our memory cells, our cytotoxic T cells, and so on. Also, these antigens are reactive. They're able to react when activated with lymphocytes and antibodies released by the immunogenic reactions. So, examples of complete antigens would be foreign proteins, polysaccharides, lipids, and things like nucleic acids. On the other hand, haptens are incomplete antigens. They are small molecules that have, by themselves, no immunogenic activity. And these are things such as peptides and nucleotides and some types of hormones. But they may become immunogenic if they are attached to body proteins, and this combination is marked as foreign by the immune system. And so these cause the immune system to mount a harmful attack. And for example, we see this with certain types of toxins such as poison ivy, animal dander. Certain detergents will cause an allergic reaction in people, as will cosmetics. So here we're talking about antigens that combine with our body proteins to make something that the immune system will react to. But by themselves, they have no immunogenicity. Okay, so just to recap, complete antigens are basically proteins that are on the surface of pathogenic organisms, and they will elicit an immune response by our B and T cells. And so our B and T cells have complementary antigen receptors that will bind to the antigens on our pathogen. And the epitope is actually the reactive region of the antigen. That is the one that binds with the active site on the antigen receptor. The epitope and the antigen receptor bind together in a lock and key fit. And this is unique to this particular combination of antigen and antigen receptors. If you have, let's say, another type of antigen, it will not be able to bind to this particular type of antigen receptor. So the way that your body is able to detect organisms that don't belong is by being able to detect their antigens. And I want you to read in your textbook about how the body actually is able to recognize all the different types of antigens that it does. In total, your body can recognize something like over 1 billion types of antigens, even though it may never have come in contact with these antigens before. And we'll talk about the reason why in just a second. So as it turns out, genes rather than antigens are responsible for determining which substances your body will recognize as foreign and react to. Basically, our lymphocytes have receptors which are coded for by different genes in the body. And even though there's only about 25,000 genes, the genetic information in these genes is reshuffled with the production of each different type of lymphocyte, so that we end up with over 1 billion different types of antigen receptors. And this has nothing to do with antigens we've encountered. It's just basically making a bunch of random antigens and hoping that one of these receptors will fit an antigen that we come in contact with in the future. So this would be kind of like going to one of those contests where they have a car and they give you some keys and say, well, if your key fits the car, you get to go home with it. In this case, your immune system hedges its bets and just makes over 1 billion different types of keys. And even though it's never come in contact with a car before, chances are statistically one of those keys will work with that car. And that's the same way that antigen receptors are able to bind and attach to antigens, even though we may have never come into contact with that antigen before. And after we bind to that antigen, the next thing we're going to do is actually replicate that cell so we have more of those different types of antigen receptors. And we'll talk more about that in the upcoming slides. So we said before that bacteria and viruses both have glycoproteins on them that the body recognizes as foreign. So these substances are in fact antigens. But we also have markers on our own cells that can help our lymphocytes distinguish between friend and foe, that is, what cells belong and what cells do not belong. And we sometimes call these MHC markers, and MHC stands for Major Histocompatibility Complex. And there's two different classes of MHC markers, class 1, which are found on all body cells, and class 2, which are found on antigen-presenting cells, thymic cells, and also activated T cells. So table 21.5 in your textbook gives a good overview of MHC antigens. Remember that MHC markers are basically self-markers that are present on all of our body cells. And MHC1 markers will be displayed by all nucleated body cells, whereas MHC2 markers are demonstrated by dendritic cells and other antigen-presenting cells. And remember, the job of antigen-presenting cells was to go out and grab a hold of an antigen and present it to either our T cells or B cells. So class 1 MHC markers are recognized by naive CD8 cells and cytotoxic T cells, whereas class 2 MHC markers are recognized by naive CD4 cells and helper T cells. So in general, if a T lymphocyte or B lymphocyte comes in contact with a body cell with either an MHC1 or MHC2 marker, it will do nothing unless that marker is also attached to a foreign antigen. For example, when cells are infected, those cells will express a little bit of the foreign antigen in addition to their MHC marker. In this case, if we have an MHC1 with a foreign antigen attached to it, it tells the cells of the immune system that I belong to your own body, but I have been infected, so kill me. Whereas we have an MHC2 marker that's also expressing a non-native antigen, it says, I belong to your own body, but I have captured a foreign invader, and so you should alert the other cells of the immune system. So remember we said before that T cells cannot recognize antigens if they're floating free in the body fluids. They can only recognize antigens once they're inside one of our body cells. We call these endogenous antigens. And one way in which an exogenous antigen becomes endogenous is by incorporation into an antigen-presenting cell. So here you can see an antigen-presenting cell that is gobbling up the antigen. This could be a bacteria or a virus. Once that antigen is internalized, a phagosome will break up the different proteins in there and digest them into smaller fragments. We will then combine those foreign antigen fragments with our own MHC2 markers. We will then post new markers on the surface of our cell, and these new markers will be a combination of our own MHC2 markers and also the foreign viral antigens. And by advertising both the MHC2 and the viral antigen, this is a way that we can alert our T cells that there's an infection in the body and that we need to deal with it. So we said before that there are B cells and T cells, and that B cells are part of our humoral response, whereas T cells are part of our cellular-based response. And T cells are actually divided into one of 3 or 4 different types. First of all, we can end up with something called a helper T cell. The job of the helper T cell is to recognize foreign antigens that are associated with MHC2 markers on antigen-presenting cells. So basically one of the jobs of the helper T cells is to activate other components within the immune system. So it activates both B cells and also T cells. On the other hand, cytotoxic T cells, their job is to destroy foreign antigens. So they're basically the hitmen of the T cell line. And we'll talk about how they kill their antigens in just a minute. And finally, we have memory T cells. And memory T cells remain behind after a cell-mediated immune response, so your immune system will be better at recognizing and dealing with an infection if it happens again. So this slide just gives an overview of the different types of T cells. So we have an immature lymphocyte that will undergo maturation or immunocompetence in the thymus. And this will differentiate either into a CD4 cell or into a CD8 cell. CD4 cells, once they are activated, will further differentiate into memory cells, which can help us to remember a pathogen, and also into helper T cells or regulatory T cells. The job of helper T cells is to activate other components of the immune system, for example, the nonspecific immune response, as well as our humoral immune response. If we go to the other side of this page, we can see that CD8 cells, once activated, will differentiate into cytotoxic T cells. The job of cytotoxic T cells is actually to destroy and eliminate infected cells in the body. So remember we said before that T cells can only recognize antigens once they are endogenous, that is, once they are inside one of our normal body cells. And so most T cells are wandering around in the inactive form, and they only become activated once they come in contact with an infected cell or a cell that's grabbed a hold of an antigen. So let's take a look at this process. So here in this step you can see antigen presentation. Here we have an antigen presenting cell, known as a dendritic cell, which will grab a hold of an antigen and engulf it. In the process, it will chop up some of those antigen proteins and present them on the cell membrane of the dendritic cell along with its own MHC2 markers. And this combination of markers lets the T cells know that there is an infection in the body. And so once a helper T cell binds to this dendritic cell, it will become activated. After the T cell is activated, it will then undergo proliferation. Proliferation is a rapid cellular division which makes clones of that initial cell. Now some of these clones will differentiate into other helper T cells, and some will differentiate into other memory T cells. Remember that the job of cytotoxic T cells was actually to eliminate infected cells in the body. So cytotoxic T cells are normally inactive unless they're activated by a helper T cell, or if they bump into an infected cell that's showing MHC1 markers along with antigen markers as well. So this MHC1 antigen complex is what's needed to activate or turn on our cytotoxic T cells. Once that cytotoxic T cell is turned on, it will destroy the infected cell in one of 2 ways. One, it may release perforin. Perforin is an enzyme that will basically perforate or punch holes in the cell membrane of the infected cell, causing it to pop or lyse. It may also release something called granzyme. Granzyme is a compound that basically initiates apoptosis in the infected cell. Remember, apoptosis is programmed cell death. So the big picture here is that cytotoxic T cells will eliminate infected cells in our body because it can recognize the combination of antigen and MHC1 markers on their membrane. Once this happens, it will kill that cell with either perforin or granzyme. Now from the last slide you might be thinking that the cytotoxic T cell is the most important cell in this cell-based immunity, but that's not necessarily true. Although cytotoxic T cells are necessary to help to eliminate infected cells, they wouldn't be able to do their job properly without the helper T cells. Helper T cells bind to antigen-presenting cells that have grabbed a hold of an antigen. And in doing that, they can activate that antigen-presenting cell so that next time it bumps into a cytotoxic T cell, that cell will also become activated. So it's a very important mediator in order to activate other components of the cellular immunity. In addition, helper T cells are also a very vital link between the cellular immunity and the humoral immunity. Once a helper T cell becomes activated by coming in contact with an antigen-presenting cell that has an antigen, it will then go out and actually activate our B cells. And once our B cells are activated, they will also undergo proliferation and start to manufacture plasma cells that will make antibodies. And so in essence, the helper T cells are very essential to the whole immune system. If we don't have helper T cells, we then have a very crippled immune system, which is what happens when we have an HIV infection. And we'll talk about that towards the end of the lecture. So now that we've talked about cellular immunity, let's go on to talk about humoral or antibody-based immunity. Remember, antibodies are produced by B cells called plasma cells. And antibodies don't actually directly kill the antigen, but they do help to incapacitate it and make it easier for it to be gobbled up by our non-specific immune response. So let's go through the steps necessary for antibody-based immunity. So first we have B cells that are binding to a free antigen. Now some of that antigen is taken into the cell and combined with our MHC2 markers and incorporated into the cell membrane. The helper T cells will recognize this antigen MHC complex and bind to the B cell, causing a process called co-stimulation. This will in essence activate the B cells. Once the B cells are activated, they will proliferate. That is, they will divide very, very rapidly. And some of these cells will become plasma cells that secrete antibodies against the specific pathogen, and some will become memory cells that will hang around for long periods of time so that we're better able to remember that specific type of antigen and deal with it more quickly in the future. So we said before that antibodies or immunoglobulins were special types of proteins made by plasma cells, which is a special type of B cell. And the antibody has this sort of Y-shaped structure consisting of both heavy and light chains of amino acids. Both heavy and light chains have a variable region which composes the antigen binding site. And so it's the sequence of amino acids in this antigen binding site that will determine what types of antigens this antibody will bind to. Now there are 5 major classes of antibodies in the body. The most numerous of these is our IgG antibodies. And the IgG antibodies comprise about 80% of the total antibody titer in the body, and they protect against bacteria and viruses. IgAs, on the other hand, are much more rare, comprising only 10 to 15%, and these are found in mucous membranes where they protect us again from bacteria and viruses. On the other hand, IgMs comprise only 5 to 10% of antibodies, and they're found mainly in the blood and lymph. And these help to activate the complement system and also cause agglutination and lysis of certain types of microbes. IgD antibodies, on the other hand, are very rare and are found mainly on the surface of B cells where they act as antigen receptors. And finally, IgE antibodies are very, very rare, and they're located on mast cells and also on basophils, and they provide protection against parasitic worms and large infectious bodies. So antibodies can incapacitate antigens in several different ways. It's important to realize that antibodies themselves don't actually destroy pathogens, but they can mark them for destruction so they can be more easily gobbled up by our nonspecific immune response. So in the next slide or two, we're going to go through talking about the ways in which antibodies actually help to eliminate antigens. So this slide shows an overview of the 4 or 5 different ways in which antibodies can help to eliminate antigens from the body. One way is through neutralization. Remember that a lot of pathogens or antigens make us sick because they actually secrete certain types of toxins. For example, bacteria can make you sick because of the bacterial toxins they secrete. So antibodies can actually neutralize these bacteria by blocking up the sites where they secrete the toxin, so they prevent the toxin from moving into the body. On the other hand, another way we can eliminate antigens is through the process of agglutination. Basically, agglutination causes the foreign cells or infected cells to clump together so that they can more easily be gobbled up by our phagocytes, that is, our nonspecific immune defense. Another method is precipitation. Here we have antibodies that bind to an antigen and make it insoluble, so it falls out of solution and once again is easier to gobble up by the members of our nonspecific defense, that is, our neutrophils and also our monocytes. And another method is through the use of complement. Complement was a special protein that can help lead to lysis of the infected cell or lysis of an exogenous antigen. And so one of the ways that antibodies help to eliminate antigens is by activating complement and causing complement to bind to the antigen. And finally, another way that antibodies eliminate antigens is through the process of opsonization. Opsonization basically means to make tasty. And so antibodies will bind to their antigen and basically attract the attention of our phagocytes, again, neutrophils and monocytes. So one of the things that makes the adaptive immune response different from the non-adaptive or non-specific immune response is we have immunological memory. That is, once we come in contact with an antigen once, we will remember that antigen and be able to deal with it more efficiently the second time around. Now, why does this work? In order to understand how immunological memory affects our ability to respond to an antigen, let's take a look at a case scenario. So let's look at day 0 and say that we've just been exposed to an antigen. Imagine here that it's maybe a cold virus. Now, if we haven't encountered this virus before, it's going to take us 5 or 6 days to come out with any kind of substantive immune response. So for the first few days of the infection, nothing's really going on. The virus is invading our cells and replicating and replicating and replicating. And so around days 6 or 7, the antibody levels or titer starts to rise. And that's because our B cells have become activated and our B cells have differentiated into plasma cells, which start cranking out the antibodies. But it takes a long time for this to occur, and peak antibody levels don't occur until around days 14 or 15 after the infection. And so, in general, it takes about 21 days to finally knock this disease down to where you're feeling better after your initial cold infection. Now let's say that on day 28 you are exposed to the same antigen a second time. Now what happened to our antibody titer? You can see that in only a few days the antibody titer has risen to much greater levels than before and much more quickly. And the result is that we knock this infection down usually before you even develop symptoms, and we clear it from the body almost immediately. So as we showed in the previous slide, once you get infected with a certain type of antigen once, you're going to be much more effective at mounting an immune response against it if you're infected with it a second time. And for this reason, we don't tend to get things like chickenpox more than once in our lifetime. And of course, there are exceptions. So one way to build an immunity to something like chickenpox is to get chickenpox. But sometimes you don't want to actually undergo the primary immune response. You want to forego the first infection altogether. And in that case, we need something called a vaccine. A vaccine is a chemical combination of dead or attenuated pathogens. And by attenuated, I mean weakened. Basically, we take some viruses and weaken them with extreme amounts of chemicals or heat, and this makes them non-infectious. But they still do have these antigenic determinants that will elicit an immune response when they're injected into the body. And vaccines are great because they spare us the symptoms of the primary immune response, but yet give us protection against that pathogen in the future. And many vaccines have something called adjuvants added to them. These are substances that will increase the immunogenicity of the antigenic determinants that we put in that vaccine, but they don't actually have any immunogenicity themselves. So in the previous slide you saw a vial of the MMR vaccine, which stands for measles, mumps, and rubella, and this vaccine was introduced in the 1960s, and you can see that it had a great effect on the incidence of measles in the United States. In the 1950s, 1960s is when we introduced the vaccine and licensed it, and you can see there that the number of measles cases dropped from somewhere around 400,000 to below 10,000. And so vaccines can be extremely effective at reducing the incidence of certain diseases in the population. But in order for them to be effective in preventing outbreaks of diseases, we have to have the majority of the people in the population actually vaccinated. So given the efficacy of vaccinations, you might wonder, why doesn't everybody vaccinate? But of course there are some social, religious, and other reasons why people choose not to vaccinate their children. So one of the reasons why so many parents have chosen not to vaccinate their children is because of a medical study, now debunked, published by Dr. Andrew Wakefield in 1998. Dr. Wakefield is a former surgeon and medical researcher in the United Kingdom. His notorious study, published in the medical journal The Lancet, suggested a link between the MMR vaccination and autism spectrum disorders in children. I should note that this study was based on only 12 children, so it had a very, very small sample size. At the time, Wakefield's results were viewed as earth-shattering. It led to steep declines in vaccination rates, both in the United Kingdom and also in the United States. And it also resulted in a corresponding rise in measles cases, which resulted in serious illness and fatalities. Meanwhile, several other researchers conducted similar studies, but failed to find any link between autism and vaccinations. It was eventually discovered that Dr. Wakefield had in fact falsified some of his data, and that he was receiving funding from consumer advocate groups who wanted to sue vaccine manufacturers. So eventually Wakefield's paper was completely retracted by The Lancet, whose publishers called the study an elaborate fraud. Dr. Wakefield also lost his medical license on grounds of academic dishonesty and ethics violations. So given all this, you would think that the vaccination scare would be over, right? No such luck. For some reason, people love to believe in hypotheses for which there is no proof. Think of all those bumper stickers that say 9/11 was an inside job, and the vaccination brouhaha is no exception. To this day, otherwise sane and intelligent people continue to ignore the advice of experts in the field, that is, medical researchers and their physicians, and instead they rely on the unfounded claims of people like Jenny McCarthy, a former Playboy bunny and anti-vaccine activist. Her son Evan was diagnosed with autism in 2005. McCarthy quickly seized on Wakefield's study and continues to promote its results to this day, despite the fact that it's been retracted. Surprisingly, she actually has a lot of followers. The end result is that vaccination rates continue to fall in the U.S., leading to an increased incidence of disease and even death in children. So if there's a moral to the story, it is that a little bit of misinformation can have tremendous and long-lasting detrimental effects on public health and safety. So just to say it loud and clear, there is no link between autism and vaccinations. So this slide just gives an overview of the different types of humoral immunity. Remember that immunity is the body's ability to ward off certain types of infections, and humoral immunity refers specifically to that immunity granted to us by our B cells and our plasma cells. Remember that plasma cells were a type of B cells that produced antibodies. And so we say that you have an active humoral immunity if you have both the antibodies and also the memory cells. For example, if you got chickenpox as a child, you're unlikely to get it again as an adult because you have lots of memory cells and also lots of plasma cells that can recognize those antigens and eliminate them very, very quickly. On the other hand, you can also get artificially acquired active immunity. This is where you get a vaccination to prevent you from getting the disease in the first place. So think measles, mumps, and rubella. So both naturally acquired and artificially acquired active immunity involves both the memory cells as well as the plasma cells. Now let's go take a look at passive humoral immunity. Passive humoral immunity involves only the transfer of antibodies. We don't have any memory cells there. So just like active immunity, passive immunity can be naturally acquired or artificially acquired. So an example of how passive immunity is naturally acquired would be when a child is breastfeeding. An infant that's breastfeeding is getting colostrum in the mother's milk, and that colostrum contains antibodies. As long as that child is breastfeeding regularly, he's going to get a dose of those antibodies that will help prevent the child from getting infections to which the mother is immune. Now, as soon as we stop breastfeeding, within a few days, the titers of these antibodies will go down, and the child will then become susceptible to those diseases. On the other hand, passive immunity can also be artificially acquired. For example, if you're in a hospital and you ever get a needle stick of a suspect patient, say that might have HIV or hepatitis, they'll probably give you a shot of something called gamma globulin. And gamma globulin is something that can ward off initial infections for a short period of time. So it can prevent you from getting the disease in the first place, but it's only going to work for a few days. If you get introduced to that disease again or have another needle stick and don't get more immunoglobulin, you'll likely contract the disease. So the big picture here is that passive immunity only involves transfer of antibodies. We don't have any B cells. Whereas active immunity involves both memory B cells, plasma cells, as well as the antibodies they create. So we usually have active immunity for life, whereas passive immunity only lasts for short periods of time. So figure 2120 on page 791 of your textbook gives a good overview of how both cellular immunity and humoral immunity works in the body. So cellular immunity is on your left-hand side, and humoral immunity is on your right-hand side. So let's take a look at step one. We have an antigen that enters the body, and we're going to follow what happens at first with our T cells. So if we go down to the first box, you're going to see that that antigen is now going to infect a human cell, and that cell is going to express both an MHC1 marker as well as part of the foreign antigen. And that infected cell may be engulfed by an antigen-presenting cell. And this antigen-presenting cell is also going to demonstrate both the foreign antigen along with its MHC2 markers. And when this binds to our naive CD8 cells, which is a type of T lymphocyte, it will activate them and cause them to differentiate into cytotoxic T cells, which will then go and kill the infected cell. It can also activate our naive CD4 cells. These are the cells that are going to become our memory cells and also our helper T cells. And so the helper T cells are very important for stimulating members of the nonspecific immune defense. Here we're talking about our macrophages, our natural killer cells, and so on. The helper T cells are also important for stimulating the B cells. That is, they help to bring in the humoral immunity. And so the helper T cells will co-stimulate and release cytokines which will activate the B cells. Just like the T cells, the B cells will then undergo clonal selection, creating lots and lots of copies of the same cell with the same types of antigen receptors. Some of these cells will differentiate into memory B cells, and the rest of them will differentiate into plasma cells. Remember that the job of plasma cells is to make antibodies, and the more antibodies we have, the greater effectiveness we're going to have at eliminating this pathogen throughout the body. We're going to wrap up this lecture talking about some immune diseases, and the first of these are our autoimmune diseases. Remember the term auto means self, so these are literally self-immune diseases where our body's immune system is attacking our own tissues. And it occurs because the immune system loses the ability to distinguish between foreign tissues and self tissues. So as a result, we start to produce autoantibodies and cytotoxic T cells that destroy our own body tissues. So now we're going to go through a few examples of autoimmune diseases. The first of these disorders is Graves' disease. If you remember back from the chapter on the endocrine system, Graves' disease is a disorder caused by hypersecretion of thyroxine, and it results in an increased metabolic rate. The reason we classify it as an autoimmune disease is that for some reason the body starts cranking out antibodies which stimulate the TSH receptors on the follicular cells of the thyroid. As a result, the thyroid starts to crank out more and more thyroxine. So even though this is an endocrine disease, its actual root is in an autoimmune disorder. Another example of an autoimmune disease is multiple sclerosis. In multiple sclerosis, the body makes antibodies which attack the myelin sheath to our neurons, and this causes a breakdown in the myelin and a loss of nerve function. It can result in all sorts of symptoms, including spasms, muscle weaknesses, pain, or even loss of limb and muscle function. Rheumatoid arthritis is another example of an autoimmune disease. Here the antibodies are attacking our joint capsules. They cause inflammation and also pain, and they also reduce the range of motion. Our next autoimmune disorder is lupus, or systemic lupus erythematosus. This is an autoimmune disease where the body's own immune system attacks healthy tissues. Symptoms of lupus vary from person to person, but include joint pain and swelling, as well as fatigue, fever, hair loss, and also a characteristic butterfly-like skin rash over the cheeks and the bridge of the nose. Lupus is much more common in women than in men. It may appear at any age, but occurs most often in people between the ages of 10 and 50. African Americans and Asians are much more likely to get lupus than other races. And finally, our last example of an autoimmune disease is type 1 diabetes mellitus. Type 1 diabetes mellitus, if you remember, results in insufficiency in insulin. And that's because we have antibodies that actually attack the beta cells that secrete the insulin. And so without the beta cells, we don't have insulin, and so we have somebody that has diabetes. And so type 1 diabetes is an autoimmune disorder, whereas type 2 diabetes results mainly because of insulin insensitivity. So how do we treat autoimmune diseases? Well, we tend to give the patient some kind of immunosuppressant drug. For example, we can give you the anti-inflammatories such as prednisone and also dexamethasone. If these aren't enough, we can actually give you more potent immunosuppressant drugs such as methotrexate. The bad thing about immunosuppressants is they tend to suppress the entire immune system, which means even though they might be very good for treating your autoimmune disorder, they're going to make it more likely that you come down with secondary infections or in some cases even cancer. Research nowadays is looking at ways to activate our regulatory T cells. Remember, regulatory T cells' job was to actually moderate the immune response and prevent autoimmune reactions. So we're hoping to be able to induce the body to actually produce antibodies against the cells who shouldn't be producing antibodies against your own tissue. It seems like it's far-fetched, and maybe it's a long way away, but we're hoping at some point that treatment of autoimmune diseases will be a much more exact process. Now that we've talked about autoimmune diseases, we're going to go on to talk about HIV or AIDS. Now, AIDS, which stands for Acquired Immunodeficiency Syndrome, is not an autoimmune disease. That is, our body is not attacking itself. AIDS, in fact, results from an HIV infection, and the HIV infection has multiple effects on the immune system, and the most detrimental of which is that it cripples the helper T cells. Remember that helper T cells were responsible for activating our cytotoxic T cells and also activating our B cells. So without helper T cells, the immune system is not going to work properly at all. And as a result, people that have chronic HIV infections are going to have opportunistic infections occurring, such as having thrush or having candidiasis, where you're having yeast growing in your mouth and also in your esophagus. The other things that can happen are things like pneumonia and a special type of cancer called Kaposi sarcoma, which is basically visible as little petechia-like spots on the skin. So unless you grew up under a rock, you already know that AIDS is caused by the HIV virus, and that HIV can be transmitted sexually through blood transfusions, through needle sticks, and passed from mother to fetus. The way that HIV cripples the immune system is by destroying our helper T cells, which suppresses both cellular immunity and also has an effect on our humoral immunity as well. Now, one of the things about HIV and AIDS that make it so difficult to treat is that it often has a long latent period. Now, this is the period between infection and actual developing symptoms. And so somebody can be asymptomatic for over 10 years. And so in this time, if they don't know they have HIV, they may be going out having unprotected sex and spreading it. Now, the symptoms actually arise when the T cell numbers drop to a certain level. And this decline in T cells causes the immune system to basically collapse. At this point, the virus can also invade the brain, resulting in dementia. We're not going to go into great detail about how actually this virus enters the cell, but it does use something called reverse transcriptase to produce DNA from its viral RNA. And a lot of the drugs that we're looking at now target this special mechanism, and they're called reverse transcriptase inhibitors. Now, our treatment of HIV today is much better than it was, let's say, 10 or 15 years ago. Now, somebody that is diagnosed with an HIV infection can usually have that infection knocked down to basically undetectable levels by cocktails of different drugs, including fusion inhibitors, integrase inhibitors, and also our reverse transcriptase and protease inhibitors. Now, there's also been a lot of research on HIV vaccines, and it's been particularly difficult because HIV is a virus that mutates very, very quickly. And so it's foreseeable that we can create a vaccine to a particular variant, but that's not the variant we're going to see 6 months or a year from now. Because HIV is such a rapidly mutating virus, it's likely going to be very difficult to develop a vaccine which will protect us against all strains of HIV. Now, before we end this lecture, I also want to talk about something called monoclonal antibodies. Monoclonal antibodies are basically antibodies of a certain type produced by a special cell called a hybridoma. A hybridoma is basically a combination of a cancer cell mixed in with a plasma cell. And it's a plasma cell that only produces a certain type of antibodies, and by combining it with a tumor cell, we're able to produce lots and lots and lots of quantities of a single type of antibody. And we use this antibody mainly in research and also in clinical testing. Okay, this slide just shows a simple schematic of how we create monoclonal antibodies in the lab. Basically, we take an antigen to which we want to raise some antibodies. We inject that antigen into a lab rat, and that lab rat will then start to crank out antibodies using its plasma cells. We then extract those plasma cells from the lab rat, and we merge them with a cancerous cell to make the hybridoma. And we can then use that hybridoma to produce mass quantities of this particular antibody. So one of the uses for monoclonal antibodies is to create diagnostic laboratory tests. For example, the pregnancy test is an example of a test using monoclonal antibodies. The specific name for these tests are called ELISA, which stands for Enzyme-Linked Immunosorbent Assay. And basically, if you've ever used a pregnancy test, you know that you pee on a stick and then you're watching for a color change. So as the urine washes down the absorbent material in that stick, it's going to bind to antibodies. And in particular, these are antibodies to HCG, or human chorionic gonadotropin. And HCG is present in the urine after you become pregnant. So if HCG is detected, it will cause the appearance of a special red or blue line on the test strip, letting you know that in fact you're pregnant. And remember, this technology all comes from monoclonal antibodies.